Coming to the first of two sessions focusing on southern resident killer whales. Wanted a little tidbit I found about the Marine Mammal Commission recently is that back in April of 1975, the Marine Mammal Commission actually sponsored what was probably the first ever killer whale workshop here in Seattle, or in general, but it was here in Seattle, held at the University of Washington. And this was back when the main issue was the live capture fishery that was ongoing in Puget Sound and in southern British Columbia. And the commission at that time was basically looking for a scientific consensus on how the species should be managed. Now, things have changed a lot in the intervening years. Obviously, live captures are not the main issue that we're here to talk about today. I will note that this population is a transboundary population. Even though it was listed as endangered in the U.S. in 2005, it was actually first listed in Canada as threatened in April of 1999. What's interesting is that I was actually commissioned to do the status review for Committee on the Status of Endangered Wildlife back in the mid-1990s. And through a variety of delays in the review process, the report didn't get finalized until 1999, at which time the population had been declining for a few years. So the initial draft actually had a not threatened listing, and the final draft, final accepted report was listed as threatened. Just 2 years later, that population, southern resident killer whale population, was listed as endangered in Canada, followed by the listing in the U.S. And I think that's important because any solutions to the problems, as well as the science that's really designed to try to understand what the problems really has to involve efforts by both countries. We do have representatives on the panel later, or a representative on the panel directly from Canada. We have one of our speakers who's Canadian, and I have dual citizenship, so I'm trying to cross both sides of the border. The program for this afternoon, for the next hour and a half, we have three presentations. We'll probably, at the end of the three presentations, we'll bring the other panelists up to the table and then have a discussion after that. So we'll probably hold questions for the presentations until the rest of the panel is up. Now, our three speakers, I'll introduce them all right now, is Dr. Eric Ward, who's a biological statistician from the Northwest Fisheries Science Center, Dr. Rob Williams, who's the co-founder of Oceans Initiative and a Pew Fellow in Marine Conservation, and Dr. Don Noren, who's a research fishery biologist at the Northwest Fisheries Science Center. And without further ado, we'll start with Eric.
Thanks. I've been working on southern resident killer whales, I think like Mike Ford said, for about 10 years now. And I think if I look back to where I started, I have considerably more white hair, in part due to this issue. Today I was asked to talk about kind of an overview of the southern resident killer whale demography and trends. And I've broken this talk into four kind of short sections. So first talking about what we know and what we think we know about the historical population size. Second, talking about an overview of the demographic summary. Third, moving on to some projections for what we think, where we think the population is going. And then lastly, closing with some data gaps. I think I've tried to really concentrate all the material from this talk on things that we've published in the past or results from previous workshops, rather than present a lot of new modeling work. So starting with how big was the killer whale, the southern resident killer whale population size before 1976, I think if you dig through the literature or even just government documents, it can be extremely confusing. We see numbers quoted as 140 or 200, and often they're cited without really without really a citation or understanding of where those numbers are coming from. And so I think, I don't really know where the 200 number is coming from. We know where I can trace back where the number of 140 came from. That was from the Killer Whale Recovery Plan published in 2008. And that was an estimate of the 1971 population size, which is kind of at the tail end of removals, plus the total removals that occurred over the last 20 years, plus a few extra animals for shootings. And so that's how you get to 140. There were about 67, I think, total removals. And I think that this calculation, is okay as kind of a first pass, but it's also potentially biased because because those removals were actually spread out over, say a 15-year period, and they didn't all occur at once. And so this has the potential for not just getting the estimate off, but for being upwardly biased from what we might expect. I did a reconstruction of the population during these 2011-2012 workshops. We know the population size basically exactly in the 1970s, and we know the numbers of removals and the ages of those removed animals based on some data that the Center for Whale Research has collected. So if we assume that the maximum growth rate of the population is, or R max is this 4% growth rate, we can back calculate what our population size would have been in 1962, and that really produces this number of something like 110 to 115. This is not the first reconstruction. I think that Dave Bain and Ken Balcom did another reconstruction of the population going back to 1960. This estimate is a little bit lower than what mine were, showing the population was fluctuating basically between 85 and 100 animals. And then the third and more recent estimate that we have is this paper that Mike Ford led in 2018, and I think we can send the paper to anybody who doesn't have it and is interested. And we have kind of two lines here for the people. I guess I should try my pointer skills, people in WebEx. We have the population size as this dashed line up here, which has been essentially flat, and then we have the numbers of breeders with associated uncertainty in the solid line at the bottom. And what that shows is that the number of breeders has also been relatively flat. Mike quoted something between 20 and 25 animals over that period, and going back further to 1970, we see that the numbers of breeders, the effective population size was also quite small. And so I think our understanding is that this population size was one that has been small for really quite some time, at least for most of the 20th century. Second, I'm going to talk about kind of a demographic summary of the population, so what we know and kind of the highlights. And we've really kind of analyzed this over and over in these 2011 and 2012 workshops. We convened also another series of workshops that were also productive up at UBC in November of last year, and I presented some other material up there. Probably the most important thing that I think we've learned in doing all this work, both DFO and NOAA scientists, is that southern residents just have lower fecundity and lower survival than other killer whale populations in the Northeast Pacific, and this translates to lower growth, as you expect. So this has all been published, and this relationship is one of these things that's really held up over the entire duration of this data set. So it's something that I think we have pretty strong confidence in saying. But what's interesting, and Mike Ford talked about this, is that really, if we look at other fish-eating killer whales in the Northeast Pacific, southern residents are really the anomaly in that they're the only one that's not growing and not doing really well. So northern residents, for example, since the 2011-2012 workshops, have increased from 265 to 281 animals. Southern residents have lost 11 animals, and so we've seen that difference. I wanted to look at what's, really since 2011, 2012, what's changed in the population. And so this is just a frequency histogram of the, we have on the x-axis is kind of our age classes, so juveniles, young males, reproductive females, old males, and old females. And I think this is, the purple bars represent the period pre-ESA listing. So this is, say, 1995 to 2001. The green lines really represent what's happened since those last workshops in this most recent period of decline. And we see that, in both cases, the results are really pretty similar. We see similar age distribution. The decline pre-listing in the late 1990s was also associated with a loss of these older males. So that was one of the big differences. And really in both cases, I think that in the most recent period included, we've seen declines in the number of old animals. And so we've lost, the biggest group that's been lost since 2011, 2012 has been these old females. This plot just represents basically the same information. This is kind of the time series of what the different fractions of the population have done over time. And the 2 points that I'll make with this is that if we, see if I can get my cursor here again, if we look at this blue line kind of in the middle, that's the old females. And we've seen them decline from, say, 12 to 15 to less than 10. And then we also see the population getting more male. So we see these older males as the light green. These are probably the males, like in Mike's paper, that are the most likely breeders or fathers. And then in kind of this light blue teal color, we have these juvenile kind of pre-reproductive males. Another problem that we've seen, and this is, unfortunately, my error bars are not showing up, but that's okay. But what we also are looking at is we see this pattern towards increasing probabilities of male births. And so, unfortunately, the data and the confidence intervals are not on here. So all we really care about, though, is the means. And what the means show is that for northern residents, over time, we see the sex ratio at birth skewed slightly female. So it's something like 47% female. And in southern residents, we see that sex ratio at birth changing from about 50% at the start of this survey in the mid-70s to something more like 60 to 65% currently. And so that's really not good for population growth. We have the potential now, I think, for the recovery and future growth to also be kind of limited by the number of females. And so in this chart, essentially what we have here are all of the females in the population that are less than age 40. These are greater than age 10 and less than age 40. So they're available to potentially be new moms, along with their age and the last birth that they had, or the number of years since their last birth. And so I think that overall this isn't that big of a problem. But I think if we start looking at these animals and looking at the age structure and how long it's been since their last birth, I think that we can start thinking that we can probably assume very little production from some of these moms. And so there's been some of these females haven't given birth in 8 to 10 years. I don't know that we can expect much more from them. I think what's the take home that I would take home from this, two things, probably that there's probably not very many females that are available to reproduce. There's probably 14 or 15. And then the second point is that I think probably the most interesting thing for me is looking at the females from K-Pod, which are in the bottom here. And so if we look at the ones that I've shaded out, these are females. There's like 4 of them that haven't given birth in about the last 10 years. And the reason that's interesting, I think, is that there's 3 of those that have basically lived through their reproductive prime in their 20s to early 30s without producing a calf. And they all are all part of the same social group. So I think it's pretty interesting to think about why they're clustering potentially in that way. And we can talk about that more during the panel discussion. Third, we'll talk about some projections for decline. And so I think that this is largely the work that we've done for these 2011 to 2012 workshops. And at that time, what we projected was the population would have this slow, steady increase. And so I think that these were our projections at that time from a population size of 87 whales. If we look at the red dot, this is basically, let's see if I can get this on the cursor again. This is kind of where we are now. That's kind of at the limit of what our uncertainty bound was. And I think in doing these projections, one of the obvious things, if you're making projections with a small population, demographic stochasticity, so just random births and deaths, is really going to drive the process and make things really hard to predict. And that's really what we saw here, that we had really wide confidence intervals about our future projections. Since those workshops, I also wanted to ask whether, we've got 5 or 6 more years of data. I wanted to ask really what's changed in terms of our understanding of these vital rates, so survival and fecundity. So we looked at how much does this new information really change our picture on these vital rates. The top 2 panels, top 2 series of rows just represent female survival and male survival respectively, and don't really show any difference. But I think what's interesting is that the last panel, the last row, represents the predicted fecundity of the population. In the red line or pink line representing the data up to those DFO workshops in 2011. The blue or teal line represents kind of our understanding after adding 5 or 6 more years of data, and we see fecundity rates come down. The reason for that is just it's not rocket science. We've had slightly fewer births than expected in the last 5 or 6 years, including those K-pod females that are not producing calves. We have, of those last 5 or 6 years, we have 3 years in 2013, 2016, and 2017 with no surviving calves produced. So that's simply why that rate is lower. In terms of our predictions, again, this doesn't include our uncertainty, unfortunately, but if you look at the 2016 status review, this plot is coming from there, and that plot does include those intervals. And what we see is that when we make assumptions, well, regardless of our assumptions about the vital rates, my projections suggest that the population is going to continue to decline, and the rate of that decline really depends on what assumptions we're willing to make. And so, in some ways, these assumptions might be overly optimistic in that I did these projections assuming a 50-50 sex ratio at birth, for example. If we let the sex ratio at birth be something closer to what it is, closer to 60%, closer to what it really is in the population, of course, the population size would go down faster. So again, we can make if we assume that these K-pod females are never going to give birth again, that would also make it go down quicker. So I think all of those assumptions are going to change the overall picture, but it doesn't change the overall trend, which is going to be likely negative. Okay, so lastly, I'm going to quickly talk about data gaps. I think this picture I just got off of the Marine Mammal Commission's website, and I think that southern residents represent kind of an anomaly. Going back to what Mike Ford had also said earlier today, we have a subset of the population of a species and an ecotype, these fish-eating killer whales that are doing really well. And so southern residents are the only one that are declining of that group. We also see overall trends and increases in many of the other marine mammals, including the pinnipeds that we just talked about earlier. And I think that if we look at all of the other fish-eating killer whales in the Northeast Pacific, we have many more fish-eating killer whales now than we did back in the 1970s. The other kind of anomaly about this population, I think, is that for a lot of these species, we know what the dominant threat or threats are and can kind of rank them. I think that something like the vaquita is a really good example. But with southern residents, I think that being able to prioritize those threats is much less clear. And so I think this is just a brainstorm that I was doing, but these are a lot of these questions I think are some things that I think are potentially unanswerable. So are the fluctuations we're seeing just due to random chance? We often don't know what the cause of these individual deaths are of the population. These animals are rarely recovered. Why are these females becoming pregnant but not producing calves? If the population is food limited, what prey is most limiting and which stocks of that prey? What season is the most nutritionally stressful? How do contaminants affect health, fecundity, or survival? How does disease impact southern residents? Are there any social behaviors? I think that transient killer whales have been documented in the last year based on some DFO colleagues there's observations of infanticide. Could potentially the same thing be happening with southern residents? How is inbreeding affecting demographic rates or inbreeding depression? And what, if anything, is the cause for these more male births? So I think I'm over time here, but in conclusion, the historical population size, I think, of southern residents has likely been small for quite some time. Southern residents are projected to decline for a number of demographic reasons associated with being a small population size, having a small number of breeding females, having this kind of depressed fecundity and the trend toward more male births. And thinking about my last slide, the relative risks or importance of these different potential factors is still a bit unclear. And with that, I'll just save any questions for the panel discussion.
Thank you, Eric. And our next speaker will be, got to get the cursor back, will be Rob Williams.
Thank you very much, Robin, and thank you very much to the Commission for the invitation to present as Dennis asked me to, really picking up on Eric's last point, what do we know about the three main threats, and how can we synthesize the best available information into some sort of population model to rank the relative importance of those threats and to identify some data gaps? I'll preface this by saying I'm presenting a paper that was published recently, was led by Bob Lacey, who invented the program Vortex, which is a great population viability analysis software. We published this in an open access journal and made a deliberate point to put all the raw data, all the inputs in the supporting information file for that paper, and would really encourage anyone who's interested in exploring this to download the paper and the PVA file itself as a starting point for discussion. I should also point out that you can see from a diverse author list that I'm presenting my own take on this paper and what it means. I think everyone in the room knows what a population viability analysis is, but one way that I think about these as distinct from traditional population dynamics models is that I think of a population viability analysis, or a PVA, as a what-if model. It allows you to explore what do we think the population might do given our best available information and our explicit statement of uncertainty. So we can vary levels of threats and uncertainty. It starts by articulating very clearly what those what-if questions are, but then it requires us to all kind of sit around a table and to do our best job at characterizing what we think is the state of the system. And so it's a great tool for just organizing the disparate kinds of information that we have about this extremely well-studied population. So that includes, of course, the basic information on demography that Eric just recapped, but it also includes what we know and what we don't know about threats. And then I think that a great application of a PVA is to run some scenarios to consider what could that population look like in future in a simple projection in the way that Eric presented, just based on projecting the demography forward, but also comparing threats and to compare management alternatives. And I think we'll learn more a little bit this afternoon about all of the wonderful mitigation measures that are in place, and we have to see, this can be a great tool to identify whether it's enough. So I think, I mean, gosh, it seems we've been going to a lot of these. This one was in 2002. This is where Aaron and I first met, actually, in 2002 at the Going conference, where, as Robin had mentioned, we'd already identified these three main threats. I mean, that was obvious in 1999. So what's changed? And I think what has changed in our paper, at least, was that we had the laundry list of the three threats, say, prey limitation. That's been well established. But things like PCBs, for example, were known to be a bad thing. But in terms of how bad and how they affected demography, we decided to take a stab at putting all three of those threats into the same currency. And that is the effect of that threat on southern resident killer whale demography. Now, we were in a wonderful situation in the sense that one of those threats, salmon abundance, how the population varies naturally, or to varying levels of salmon abundance, we have the gold standard data, thanks to Ken and the analyses that John Ford and Eric Ward had led. And as Eric pointed out, that we knew that as salmon levels dropped, we saw demographic consequences in all age sex classes. And so we know that salmon abundance can be linked to killer whale mortality, but it can also affect reproductive rate. For the other two threats, noise and contaminants, we were in a little bit tougher situation in the sense that we had to quantify that threat and how we think it affects demography. And the way that we got around that for noise was to say, let's just include that as a multiplier that says we can randomly vary the amount of salmon that's available in the environment and then sequentially reduce that proportion of that salmon that is accessible to the population. And this is based on some work that we've done to show that the whales spend 18 to 25% less time feeding in the presence of boats than in their absence. For the contaminant side of things, we really drew on the wisdom of Ailsa Hall, who had just been working with Gina Ilatalo and Lori Schwacke and others to try to take what we know about bottlenose dolphins and mink and other mammals to try to predict a PCB tissue concentration level to a demographic consequence, the probability of a calf surviving. And so it's a bit ugly, but we did at least try to make, rather than an apples to oranges comparison, at least tried to make them all apples. Some of those are easier than others. Now, in terms of how you can use a PVA to ask what-if questions, of course, we only have the one population, but what you can do, of course, is run these individual-based, agent-based models and simulate, draw from the observed demography, and then have that system perturbed by what you think you know about these three threats. That allows you to run tens of thousands of simulations and to use the model to explore how the population might proceed in terms of status quo, which is the same as, effectively the same as Eric's forward projection without additional information on, that's assuming that the threats remain constant. We can consider a scenario in which those threats increase in some way, and I think with noise that is certainly a possibility. With what we heard this morning, the condition of salmon worsening over time is certainly a real possibility. But we can also use this to compare and contrast potential options for mitigation of these three threats, singly or in combination. And so I think it's a useful tool for even just for stimulating discussion between scientists, among scientists, or between scientists and stakeholders or managers. So I'll start with that status quo example. We started our model based on demography up to, I think, 2014, which may explain why our baseline scenario was slightly pessimistic, slightly less than 1% annual decline, but effectively a stable or slightly decreasing population size based on the observed demographic rates between 76 and 2014, given no additional information on changes in the threats. Now, of course, in the real world, we know that threats will not stay constant, and so this is a great way to think about asking those, what are the factors that are affecting the population as it is now? And so we took the three threats. For salmon, that's, as I said, that's a fairly straightforward one. We can look up the value of the salmon index on the various indices that Eric and others have talked about. For the others, we took a plausible starting point, but then we had to iterate to try to make sure that the baseline level, what we were calling the current status quo level of each of these threats, of noise, the effects of noise on foraging efficiency, the effects of contaminants on calf mortality, resulted in a population that mirrored the demography over the last 40 years. So that became our baseline. Fortunately, the model doesn't hinge on getting that baseline exactly right, because what we're interested in future steps is comparing the relative importance of each of these threats. So it's a plausible starting point for discussion. But I think it does say that the status quo is not going to get you to the stated management recovery target, which here is, we've just plotted the 2.3% growth rate. For comparison, of course, Canada does not specify a quantitative recovery target in the same way, or at least initially it had not specified a quantitative management goal or conservation target. But just for your own interest, we've marked a stable population down here and the 2.3% growth rate here. And the starting point, of course, is predicting a stable population, but we want an increasing one. What this shows you here is that if you could somehow wave a wand and increase calf or decrease calf mortality, increase calf survival by 50%, get rid of half of the calf mortality, you would get an increase in population growth, but that's really not what's driving the population. If you could do something about adult mortality, you get greater population growth. But in our models, at least, it was fecundity that we really wanted to, that would have the greatest impact on the population growth rate. This is given our pre-demography links, remember we're conditioning on the Ford et al. Papers and the paper by Eric in 2009 for fecundity. If you imagine, this is a little confusing, isn't it? If you imagine here at the 50% rate, that's about our starting point for discussion. This is effectively our straw man population viability analysis, our placeholder value for what we think the current levels of the threats might be. And so what we say is, if you can then imagine, if you could somehow alter the availability or the abundance of salmon in the environment and adjust that parameter alone, then you could imagine if you could get back to the biggest years, the best salmon years that we have on record since the 70s, that would get you up to a population growth rate approaching 2.3%. So that alone would make a difference. Whereas if we get back to the worst salmon years on record, we would have a population decline of almost 4% per year. Contrast that with what we think we know about the effects of PCBs on calf survival, and we see that, here in the middle, PCB concentrations alone are not causing enough calf mortality to explain the current, position of the population. If we could somehow make that problem go away, that alone is not likely to give you much of an increase in overall population growth rate. And even, I guess the flip side is that even if it got worse, if it got twice as bad as it is now, 2.5 times as bad as it is now, that does not cause a major population decline. If you could somehow make noise better, if you could mitigate the effects of noise, that could get you, given no additional information on salmon, that could get you up to 1.5% growth rate. So it's a starting point for discussion. But it really does help organize your thoughts and focus a conversation with stakeholders and managers about how you could potentially use this to inform an effective mitigation or action plan. And again, this is maybe a more intuitive way of plotting the same data, but if we could improve, so this doesn't look at worsening situations, but if we could improve the PC-based situation, we'd get up to about 0.5% growth rate. If we could reduce noise and disturbance, we think, across the range of data we considered, we could get the population growth rate up to about 1.5%. If we got up to the best years on record for Chinook salmon, back in some of the best years that we've seen since 1976, we could approach that 2.3% population growth rate target. But what was interesting was to consider a 50% decrease in noise in addition to a 15% increase in Chinook salmon abundance. And if you could somehow mitigate these threats in combination, if you could do something to improve the abundance of Chinook in the environment and increase the proportion of that Chinook that is available to the whales by reducing noise and disturbance, that double mitigation approach causes a benefit to the whale population that's larger than you would get by treating any one factor alone, even under the most optimistic scenarios. So as we discussed, among the co-authors, I think we all agreed that it is a perfect storm of threats, but we shouldn't lose sight of the fact that Chinook salmon are at the, well, we now know this morning that salmon are at the eye of the storm. So the whales do need a new normal. They need abundance that is something that we haven't really seen since the 80s. We can't lose sight of the fact that oil spills, are a real threat. We did not model those explicitly in this, but of course the framework is available to do so. And so what we do need is that a very large proportion of that newly abundant Chinook salmon to be available to the whales. And so recovering salmon while reducing noise and disturbance will accomplish more than addressing either threat on its own. So I think this is a useful tool for exploring the what-if questions. Where do we think we are along that range of data? What would the consequences be at the extreme values of those threats that we considered? But we really could then use that to focus a discussion about where do we think we are across that spectrum of each level of the threat. With respect to salmon, as I said, that's fairly straightforward. For noise, we need better field data on linking the foraging efficiency of the killer whales as a function of noise. For contaminants, man, I wish we could have included PBDEs and some of the other contaminants of emerging concern. I wish we could have considered indirect pathways of effects, say, PBDEs making animals, causing immunosuppression that causes disease outbreak or something else that was, for which we didn't have any data. But the limiting factor here is that you have to have experts who are willing to quantify a link between the threat and the demography. Currently, we can't treat these in qualitative ways. So other pathways of effects would be a great thing to add to this. Shortly after we published this paper, there was that fantastic report out on population consequences of multiple stressors, and I wish that we'd had funding or capacity to start to think about how some of these threats could be working together synergistically, antagonistically, or in an additive way. And I think, again, now that there have been some real-world proposals out there for mitigation, this might be a useful way to consider the population consequences likely to arise from that mitigation, given the available information. So, of course, it's important to separate the science from the policy. It's important to separate the what we found from what do we do with the information. You won't be surprised to find that even among our co-authors, we disagree about what this information should mean for informing real-world mitigation. It would be wonderful to take these last 5 years of data, a few years of data, to use the model to predict how the population should have behaved, and then test that. But I'll reiterate that point that the science is open access. You can download the PDA file from Bob's Vortex website. And I'd like to maybe end on that note that Bill McKibben said about climate change, and that there are no silver bullets, there's only silver buckshot. And I think what we found in this exploratory exercise is that it will take mitigation of a couple of threats together to get us where we want to go. Thank you very much.
Thank you, Rob. So our next speaker and our last speaker before we go to the panel discussion is Don Norn from the Northwest Fisheries Science Center. And just a sec, sorry.
Thank you very much. This is a perfect introduction that Rob just gave me, because I'll be talking about a kitchen sink full of issues and trying to make connections with some of these potential issues for cumulative effects. And so, as we've heard from everybody up here before me, there's 3 main key risk factors: lack of sufficient prey, which could be quality or quantity, disturbance from sound in vessels, and then the chemical contaminants. And also, Mike Ford mentioned, as Eric has as well, that we have another potential issue that should be considered a fairly strong threat, which is the small population size. So my background is physiology, and I've done a lot of bioenergetics work in particular, and have branched out into a lot of the other anthropogenic impacts. And so I'll be talking a little bit about bioenergetics to start, because I think it's a great tool to really understand assessing different impacts, in particular the impacts of reduced prey availability, and also the biological significance of the behavioral responses to disturbance and the interactions that you might see from those two. And I'm sure everyone here is aware of this, but just as background, basically, in the bioenergetics math, we can kind of look at it simply by just equating energy in to energy out. And if you have a deficit, you can't grow. You may have deaths or poor condition or susceptibility to disease. If you have an increased energy input, then you have the potential for growth or reproduction. And if you're just status quo, you may remain neutral. So I'm mostly going to talk about work that's published and kind of put it in a framework of interacting effects, and just to highlight some of these studies. And so we've all heard a lot about the Chasco papers, and just one of the confounding factors with these prey issues is that there may be some competing predators. It's also interesting, this group of animals is highly social, and there's been a couple studies that have highlighted how these social dynamics change with, and that includes habitat use patterns, group size, cohesion, which changes during periods of decline as well as periods of prey availability changes. So there tends to be, across these different studies, evidence that suggests that there's smaller southern resident killer whale groups traveling together during periods of decline. Also, greater movement, reduced presence in their core habitat, less connected social networks, and smaller groups during periods of low salmon abundance. And on an energetic standpoint, that makes sense because it reduces competition among conspecifics for prey. But these are social animals, and there's benefits of sociality, such as prey sharing, calf-rearing assistance, and reproduction. So there's been a lot of studies looking at these prey issues, and one of the studies that has come to the forefront is looking at fecal hormones, and those results suggest that there may be inadequate prey in some years. But they also pointed out that vessels may also contribute to southern resident killer whale stress, particularly during periods of low prey availability, and I'll be talking about the potential impacts of vessels during low prey years. Jessica London also recently published a paper that showed that fecal persistent organic pollutants tended to be higher when prey abundance is low, And this could indicate that these animals have higher circulating POP levels when they're food limited, or they could be prey switching, either way. But basically, there is a potential risk when these animals are not meeting their energetic demands that they're freeing up some of the contaminants in their blubber that may be higher in circulation and is then evident in the fecal material. Also, there has been a lot of focus on photogrammetry methods by John Durbin, Holly Fernbeck, and Lance Barrett-Leonard. And they've looked at changes in condition. They can see this through aerial photo images, and it's possible that these animals are also losing condition during periods of reduced prey availability. Mike mentioned that there might be some seasonal differences that have been found. So that's not a terrible figure. I didn't make the figure. I'm not going to, it's very busy. So let me just explain. I'm going to talk about vessel impacts now and behavioral and physiological effects. And so one of the things we were asked is basically, how are things changing? And so Soundwatch has been monitoring the vessels around the whales for a long time. And so this is their plot. And the main thing to see is that this plot extends from I can't even read it, basically 1998 to 2016. And you can see that the black line is maximum vessels around the whales, and there's some averages both for commercial whale watches and also recreational boaters. And you can see that these animals have a fairly high level of boats around them, and it has been pretty much consistent over the last couple decades. There's been a lot of studies looking at behavioral effects, and this is just a summary that there have been a lot of ephemeral behavioral changes that have been noted, including changes in respiration rates, swim speeds, path directness, and also increases in surface active behaviors when there's more boats around or when they're closely approached. Also, there are vocal responses by the animals. When there are more boats around, the background noise is higher, and they increase their call output. One of the potentially major issues, though, is that there's a decrease in foraging behavior and increasing travel when these whales are with boats, as was mentioned previously. And as I mentioned before, vessels may contribute to stress hormones, particularly when prey is limited. So we have looked at some of these energetic impacts of these behavioral effects, because it's kind of ambiguous when you mention that there's a behavioral effect. How significant isn't? So a lot of the efforts that I've been part of recently has been looking at the energetic costs. And so the energetic costs of these ephemeral behaviors are fairly low. Also, the energetic cost of modifying the activity budget is relatively small, as Rob and co-authors published earlier. And I wanted to show the figure, and Rob had mentioned this as well, the change in activity budgets when vessels are near versus when they're not. So you can see on the left panel that they are significantly reducing their foraging and increasing travel. And I used Rob's previous methods to add on to his data from northern residents to show as the animals are surrounded progressively by boats as a number of hours per day, that you have a significant reduction in foraging behavior, which he had mentioned before was fairly substantial. So the other major risk factor are the southern resident contaminant levels. And basically, this is a fairly old figure. It would be great to see some recent data, but this just compares 3 of the killer whale populations that are found around this area: northern residents on the left, southern residents in the middle, and then the transient mammal-eating whales on the right. And the dark bars are males, and the lighter bars are females, and you can see that southern residents have higher contaminants compared to northern residents, and across the board, females, because they're offloading, tend to have lower levels than the males. It's interesting to note how contaminated transients are, but they seem to be doing okay. So it's an interesting area of study. Oops, sorry about that. So this figure is from Cron et al. 27, and just shows different ratios of the different contaminants. And you can see there's J-pod individuals on the left, L-pod individuals on the right. And what it shows is the relative ratios for DDTs compared to PCBs in light gray. And you can see that L-pod individuals tend to have higher DDTs. And then compared to J-pod, which tends to have the higher ratios for PBDs and PCBs. And that's because these animals, these different pods tend to travel differently. The habitat use patterns, particularly in the non-summer months, is quite different. So the L-pod individuals are the ones that go to California. The J-pod are the ones that are found more in our local waters more often. So their habitat patterns will predispose them to different risks based on what they're eating and what they're exposed to. I recently got this from Gina Yolatalo that shows the temporal trends from May 2005 through September 2017 for males. So you're basically taking out the reproductive effect. Looking at PCBs and DDTs, there is a slight but significant decline. There are some potential things that should be looked at, such as age of males and the pod makeup, but there might be a declining trend in these contaminant levels. There doesn't seem to be much of a change for PCBs over this same period, though, or PBDs, sorry. So we, just to remind people for the contaminant risks, we have POP exposures can be linked to immunosuppression, endocrine disruption, and reproductive failure in marine mammals. And there's been a recent model that Rob had mentioned from Elsa Hall that looks more at calf survival as well. POP exposure can also interfere with developmental processes, which has been found in lab experiments on mammals. And this is particularly key for developing neonatal animals, neurological effects that can be exacerbated by having multiple contaminant exposure, PCBs and PBDEs. So we do have an issue where POPs may be decreasing the reproductive success and survival. So it's also likely that this risk will vary across females based on their reproductive history and body condition as well, and I'll talk about that a little bit. So I've been looking at milk and serum from killer whales and dolphins that are trained for collection of blood and milk. And so we have found that the primiparous females, not surprisingly, have higher serum and milk POP levels than multiparous females. And this is what you would see in the field. Gina published a paper that showed that the firstborn calves tend to have higher levels even as adults. What is interesting is that we found that the females tend to significantly offload these contaminants early during the lactation period. So there is a precipitous drop in both milk and serum levels, her serum and her milk levels in the first couple months. So the firstborn calves are then exposed to higher POPs and for longer periods of time. Basically, the females that are multiparous have a shorter amount of time before they asymptote off and they have lower levels. So the firstborn calves are exposed to these higher levels for a longer duration. And so they might be at the highest risk. And it's interesting, if you look at some of the literature from the pinnipeds, Kathy Debier published a study on it's fasting elephant seals, but I think, physiologically it's valid. She found that leaner elephant seals had higher circulating POP levels, and as they fasted for longer periods, their circulating POP levels also increased. So I think we could probably equate this to a potential higher risk during prey limitation, which kind of goes hand in hand with what the fecal material showed with higher POP levels during prey limitation. So to pregnancy loss, John Durbin and his colleagues have also been looking at pregnancy loss with photogrammetry, and there seems to be a high rate of reproductive failure. They're able to detect pregnancy within 4 to 5 months. Sam Wasser has also published a study that shows the same thing using fecal hormones, a fairly high rate of reproductive loss, and particularly some of these are during late gestation or early postpartum before a calf is observed. So this is a fairly expensive time to be losing this calf, and one example is J32. The other thing that's been investigated with the photogrammetry technique is condition. They've found for the last 5 out of 6 southern resident killer whale deaths that those animals showed a decreased body condition, except for J34, which was killed by blunt force trauma, most likely a ship strike. And also they had documented other deaths, other changes in condition prior to death with their helicopter work as well. And the recent paper published by Holly Fernbeck stated there were some declines of body condition for some animals from 2008 to 2013. The question remains, is this due to disease or prey limitation? So this is a busy figure, and I'm responsible, so sorry about that. So there has been a standard necropsy protocol for killer whales, and these are data from all killer whales in the Pacific, not just southern residents. But just to kind of understand what any relationships between cause of death and condition, I did a simple body condition assessment of just the girth in front of the dorsal fin divided by body length. Animals that are older have bigger body length, and then more robust would be animals that are fatter. So the top animals we can just delete because they're either pregnant or bloated. But it is interesting to note, if you look at some of these more robust animals in the blue bar, the majority of them are from trauma or ship strike. So there seems to be some indication that animals that are struck by ships or have trauma have a better body condition, as you might expect. What's interesting, though, if you look at all the poorer animal body conditions, these are due to either starvation or emaciation or disease. So it's very difficult to even tease apart some body condition differences with just cause of death. But it's important to note the animals that are diseased and animals that may be prey limited are going to potentially show poor body condition. And we do have a fairly low recovery rate for dead killer whales, so this will limit our ability to assess, all life-threatening risks. So I am running out of time. The other thing that I wanted to touch on is health. There's been some work looking at breaths from killer whales, and I wanted to point out that they have found bacteria that is resistant to antibiotics. There have been fungal infections that have been found in a southern resident as well as in Patrick Harbor porpoises that are detrimental. And then just the small population size and their social behavior, communicable diseases have potentially could have a large effect on these animals. And I want to just highlight some direct human impacts. There have been, in all the necropsy reports from all the killer whales, some fisheries issues. Southern residents, so far, all they've seen is a flasher. But I did want to point out that a northern resident did have a cause of death that might have been linked to a fisheries interaction, and there are some other animals that have been entangled. The other thing that is important to understand is that there is some direct human impact that might not have been noted as important previously, and that includes ship strikes and blunt force trauma. So we've had 3 southern resident killer whales die of either blunt force trauma or ship strike. One was a whale that had affiliated itself with vessels, but recently the J34, which is on the bottom panel, had a huge hematoma consistent with a ship strike. And then there was L112, which was a blunt force trauma of unknown origin. And then there's other northern residents, offshores, transients that have had ship strikes. So the point here is that there's other impacts that really could be important for especially a small population. If you knock off one whale, a year from a ship strike, then you have a decline. So Mike also had mentioned there's other issues such as potential inbreeding effects. An example from his paper showed that 2 males have sired 52% of the progeny born since 1990, so there's potential inbreeding depression. And then they have different habitat use patterns, as I mentioned before, so the relative effects of the different risk factors could differ across the pods. And in conclusion, these animals face several risk factors, and then additional threats have been identified, but we aren't really sure of all the impacts of all these additional threats, which has been mentioned before, or the interactions. It's really difficult to tease these threats apart, as Eric had mentioned, because some of these threats could be increasing or decreasing together, which makes it difficult to tease apart. And there are potentially additive effects. For example, the vessel impacts on foraging. The DTAG work that the Northwest Fisheries Science Center is conducting is trying to look at this foraging efficiency question with noise. And then we also have a really difficult time understanding these contaminant effects on health and reproduction. And then obviously I just mentioned the direct human impacts. So that's it. Thanks for your patience.
Thanks, Don. And I think what we'll do now is we'll have our panelists join us up at the table here in front. If you guys would all like to come up. Then we have, I'll just go through who's going to be on the panel as well as our 3 speakers. Mike Ford, who we heard from this morning from Northwest Fisheries Science Center. Ken Balcom, the founder and senior scientist with the Center for Whale Research. Gina Ulatalo, the Environmental Chemistry Program Manager from Northwest Fisheries Science Center. Dr. Sheila Thornton, research scientist with Fisheries and Oceans Canada, Dr. Jason Woods, the operation manager and senior research scientist at the Sea Mammal Research Unit Consulting, and Dr. Joe Gatos, the senior wildlife veterinarian and science director with the CDOC Society. And I think what we'll do with the questions and the discussion is we'll start with the Marine Mammal Commission and commissioners, committee scientific advisors, and staff. And then if we've exhausted those questions, we'll go to the rest of the room later on. So anyone has a opening question? The other thing I'll do is I'll put up some topics of discussion that we could consider. And I will also mention that the next session following this is a management section, so we can keep that in mind in terms of how much we focus the questions on questions and discussion on the science versus on the management side of things. Peter.
I've got a quick question for Dawn, which is, well, but you have I think Rob or Eric showed the long period between calving in females, or the long lag since their last calf. Do the persistent organic pollution levels build up if you don't have a calf for 14 years and then you try to have one somewhere in that period, like a primiparis mom?
Right. I think we can try to tease that apart from some of the biopsy data, but also with the whales and the dolphins that I've looked at, you can definitely see a difference. So I've had the fortune of having different females with different reproductive histories, and one of the dolphins, for example, had been she was a little older, but she had spread her calves around, several years across, and so she was almost as contaminated as the primiparis dolphin female that I looked at. So for sure so the more time they go between calves, they have the ability to build those contaminants back up again.
Oh, and I should also say that if any of our panel members want to say anything in response to any of the presentations or anything relevant to the earlier presentations, or then please go ahead as well. Joe?
Yeah, Joe Gatos. I just wanted to point out one thing that didn't come up in the presentations, and you're probably all thinking, but I just want to say it out loud, is that the time between births is not necessarily the time between births. And I think all of us could probably talk about some anecdotal evidence on that, but there's probably animals that are born and never seen again, and we do see some evidence on necropsies of animals that we thought were primiparis, but may actually have had a CA, had a birth before and then not had that birth. So just want to point that out.
Francis?
Well, first, I'd just like to thank everybody for the presentations, and I think some of you know that I was involved in the panel with the Hilborn report, so it's great to see how much work has been done since then, and how hard it is to tease out these interacting factors and the cumulative impact. So one area I was curious about is how much use has been made of the comparative northern resident population. And maybe looking at Sheila here, but I haven't seen much on, for example, comparing fecal stress levels to individuals that perhaps do share habitat at certain times of the year. And could more be done there? Because we're struggling with such a small population that it seems that they might be a useful approach. So I'm just more of a sort of general opening question on what we.
Well, we completely agree on that, and the northern residents are such a nice control population because the numbers are up at 309, and they're foraging on relatively the same or similar stocks that are coming down. So this summer we are initiating a couple of studies that are direct comparisons between the northerns and the southerns. We'll be collecting fecal samples to look at the stress levels between those two different populations. We also have a collaboration with Marla Holt and I don't think she's here today, and Brad Hansen. They will be doing DTEG work on the southern residents, and we will be complementing that with northern resident DTEG work. And this will be nighttime DTEG work. So we'll try and establish the foraging behavior at night, which we really don't know much about. So this will lead into some mitigation hopefully some proposed mitigation for the populations. So we are absolutely focusing on the northern residents as a very good example of a controlled population.
Jason?
Following up on Francis's comment, I was kind of wondering specifically about comparisons with the contaminants, and you did a PVA where you through expert opinion, it sounds like came up with a relationship between vital rates and contaminant levels, and did some projections about the effect of that on the growth rate. But that's pretty dependent on those functional relationships that you assume. And I wonder, what do we make, and maybe this is for Gina, of the graph where we see much higher contaminant levels in the northern residents, and yet the population growth is fine.
I think those were transient.
Oh, you have to hold the.
I think those were transient whales that had the higher contaminant levels, but the Canadians are also doing contaminant analyses of recently collected biopsy samples. Peter Ross is working on that, so we will be able to start doing those comparisons. I don't have any recent data on transients. I think the last transient killer whale samples that we've analyzed were collected in 2005, so it'd be great if we could get some additional data on the transients to see how their contaminant levels are comparing now, because that's over a decade. But I still think that their levels will be higher just based on where they're feeding in the food web. They'll be higher. I would like to start looking at patterns of the contaminants. Do the southern resident killer whales have higher proportions of higher molecular weight PCB congeners compared to the transients? Because what they're finding with bottlenose dolphins down in Georgia is that they seem to have these animals that are exposed to these higher chlorinated PCB congeners seem to have more health issues associated with exposure. It's an association. It's not cause and effect, but and we haven't really teased that apart yet, but it's something to look into.
Sheila.
And Francis, back to the northern and southern resident comparisons. During the fecal collections, we're working with Lance Barrett-Leonard as well, and he will be undertaking epigenetic analyses to look at up and down regulation of known markers that respond to contaminant loads. So we'll be taking that angle as well.
Actually, I have a question about the epigenetic analyses as you just outlined. I've heard of that being used in terms of aging animals, but I hadn't heard about it in that context. I don't know if you could just expand a little bit.
I would confess my absolute ignorance on genetic topics at this time, so I'm not going to say too much about it, except that Lance has done, he's publishing a paper coming out shortly, and just looking at up and down regulation of particular components.
Mike?
Yeah. Just a factual question. Try to understand. Do both southern residents and northern residents feed upon Fraser River Chinook salmon? In the same proportion?
I don't know if it's the same I don't know if it's the same proportions exactly, but there is a broadly overlapping set of stocks that they do feed on based on the diet information from DFO.
How are Fraser River salmon stocks doing?
Generally not as well in terms of trends as Columbia stocks.
How about in comparison to other Puget Sound stocks?
The estimates we have overall is that there's been a decline of about 15 to 25% of Salish sea salmon altogether. Fraser River spring stocks have been particularly down. The summer runs have actually been up, particularly the sub-yearling summer runs, and the fall runs are also down. So it's a bit of a mixed bag, but overall there are a number of stocks in the Fraser that are a source of some concern. I
have actually just one sort of comment for Dawn. You mentioned that the animals that were ship-struck were all relatively good body condition, and you just wonder whether or not, I don't know if anyone's done any modeling of at what body condition the animal's actually likely to float and be found, and if any of those animals that are nutritionally stressed or ship-struck, whether they would just sink and never be found, and particularly given, I don't know what the estimates are of cryptic mortality in killer whales, but maybe Rob or Joe could comment on what proportion of the animals that die are actually found.
Yeah, Don pointed out in that Barbieri et al. Paper, we looked back and there was 20% of southern residents that were found and 3% of northern residents that were found known dead animals. And that's a great question. We have no idea. I mean, we all have bogus hypotheses, or maybe they're not even hypotheses. Maybe they're just guesses that, there's a possibility that thin animals could sink and not make it to the beach, but I don't have any data to support that.
Peter?
Yeah, going back to Rob's sort of multi-phased approach of dealing with noise and foraging, you're mentioning you're doing nighttime detagging. Can you compare daytime and nighttime in terms of presence of vessels around the whales? I mean, would that be an accurate comparison given that it's day and night? And then the other question is, between northern and southern, have you done the comparisons of foraging efficiency with relative numbers of vessels?
That's exactly what we're undertaking right now. We do have the daytime, excuse me, the daytime DTAG data. So we're doing the northern-southern comparison. We can make a reasonable assumption that the majority of whale watchers are at home at that time of night. And we can then quantify the presence of the commercial vessels through AIS, or Automated Information System, so then we can figure out what the relative pressure is around the animals in the daytime versus the nighttime. And then we'll add in, over the next 3 field seasons, we'll be collecting the DTAG data at night and do that comparative study between the northerns and southerns, given the vessel pressure, acoustic environment, and then received sound. And then the foraging, and then bring in the variants of the foraging behavior and hopefully catch per unit effort. We're going to see if we can tease that out from the DTAG acoustics.
And one comment on the detagging work is we actually have a couple hundred hours of time depth of quarter data from southern residents from the late 1990s and early 2000s. That would be great to throw into the mix for the comparison to see whether there may have been a change in either activity levels at night between those 2 time periods, and add to that study. So. Francis?
So, a question for Joe. I think Dawn did a great job of pointing out these sort of, at such a small population size, how we're dealing with individuals, and some of these are stochastic events like, a ship in the night can make a big difference. So I know you were working on a sort of individual animal database, and I was just curious about the status of that and how it's doing.
Yeah, good question, Francis. So we have we put together an access-based database with National Marine Mammal Foundation, and Ken's put in all the data from the different animals, and we're uploading all of the necropsy data for that. Gina's putting in the contaminants data. So we're just in that process of Dawn's putting in data from the morphometrics, from the necropsies and things like that. We're just at this point right now where we probably have enough stuff that over the next 6 months we can start cross-examining some of the data sets, but it's been a little bit of a slow process. But I think it has high potential on 2 cases. One, at looking at some complex interactions, and I think it also has some value to have individual animal health data records. So if we can get in photogrammetry data, sighting data from a lot of different researchers, then if something were to come up with individual animals, we would be able to look at that and say, I think when you get a small population, southern resident killer whales, mountain gorillas, things like that, you start to look at individual animals differently than you do with really large populations, and I think it could be a big asset at that, for that perspective as well.
Ken?
Talking about individual animals again. I'm just a cowboy zoologist from New Mexico. Came here to sort of evaluate the herd. And I really appreciate the mathematical work of my colleagues here, Rob and Eric, and the genetic work of Mike Ford and Kim. But there is a lot of information available from all this observation, 8,000 encounters with killer whales in the past 40-some years. And, you know, like, we can actually pick out which ones are the breeders. And you've got one living male that has produced living offspring. That's L41. That's all we've got right now that we know of. And we only have 5 females that have produced calves in the last 5 years, which used to be the average calving interval for almost 30 individuals. So we definitely have something going on. And I think we can dissect more of the individual histories of these and provide some, at least, insight into some of the hypotheses. I think we could answer 3/4 of the questions that Rob posed in his last slide.
Dennis?
Yeah, I wanted to follow up on that, Ken. So if K27 was lost, would the K-pod be reproductively dead, or are there subadult females that would eventually hopefully reach reproductive age and provide the possibility of resurrection?
Let's see here. In my back pocket here, we have one subadult female K43 who potentially in the next 10 years could be reproductive. So the answer is one possible if she survives. K27 is basically it. And, you know, K-pod is flat. They haven't been doing anything for 15 years.
Yeah, I think it's important to remember the historical perspective because, you know, back in the day, L-pod had 54 whales, and L-pod has been declining pretty steadily for a long time, and most of the recovery or most of the increase among the pods is all in J-pod, and so what that implies to me is that the factors that are influencing the dynamics, population dynamics for this population are acting separately on different pods, and so knowing more about what those different sources of either benefits in terms of are they spending more time in areas where there's higher salmon concentrations or wider seasonal diversity of salmon concentrations, or what are the other threats that are facing L-pod in particular.
Yeah, with respect to L-pod, there's 10 young males and 4 young females. And there are probably of the 1, 2, 3, 4, 5, 6, 7 or 8 matrilines around, half of them are dead and going nowhere. And there's only 2 of them that have young females that might come into the reproductive cohort. So it's not looking too good for L-pod.
Tiff?
I have a question for Jason, who I can't really see over Randy's head, but I know you were involved last summer with the ECHO program off of Vancouver, so could you give a very brief synopsis on what the results were from that study?
It's already on. Yeah, Tiff. Yeah, maybe Robin, if you go to that slide there. Yeah. So the ECHO program of the Port of Vancouver led a ship slowdown study in Harrow Strait. This past summer was a voluntary 2-month slowdown trial from more than 2 years of acoustic measurements in Harrow Strait at Limekiln. We know there's a lot of variability in background noise levels, and the average noise levels are pretty loud. And oddly enough, on average, the loudest periods that we have at Limekiln are actually driven by small boat noise, not by ships, but ship noise comes in a second at that. And that's because Limekiln's about 2.2 and 5 kilometers away from the inbound and outbound shipping lanes to the Port of Vancouver. So during the slowdown trial, 56% of the piloted vessels, so not all large vessels are piloted, but of the piloted vessels, 56% reduced their speed to less than 13 knots. So this was a pretty good chunk of the vessel traffic, especially given that this was a voluntary slowdown. The map on the left there shows you sort of the polygon that was the it's about a 16 nautical mile long corridor. While the source levels of those ships reduced significantly, so the initial numbers that I've seen so far for, say, reduction in source level of some of the faster ships, like container vessels and passenger cruise lines, were on the order of 10 or 11 dB. By the time you combine all of the different ship types and the distance to Limekiln from those different those two lanes, shipping lanes, we found about a 2.5 dB reduction. So it's still a significant reduction in noise levels that can be attained from this type of mitigation. And obviously, because ship noises tends to be concentrated in lower frequency bands, we find the largest reduction in the first decade band from 10 to 100 hertz.
Mike?
Right. So you are the 9 experts sitting up there. You are the science experts. Is the science that is required to sort this out being done? Or is there something else that should be done in your view?
I think one of the things that I think should be done, and I think will be done now in cooperation with Canada, is some more of these detailed comparisons of the northern and southern residents. I think if we knew with some certainty what was causing the northern residents to grow and the southern residents not, we would sort of have the answer to our problem, at least from a scientific point of view. You know, the problem all along has been that there's so many there's so many differences that teasing out what those are has been very difficult. So I think that some of the DTAG work, quantifying, are the northern residents actually eating more salmon, for example? Are they more successful at preying? Will help us evaluate the prey hypothesis. Are they significantly less exposed to noise and therefore spending more time foraging? Will help us evaluate those hypotheses. I think that'll be very helpful. I guess from my perspective, though, one of the things, with quite a bit of scientific investment over the last 15 and really 30 years, 40 years, I think one of the answers that we might just have to accept is that it's pretty complicated and that, there may not be a lot of super easy solutions to this problem.
Sheila?
Excuse me. In keeping with the principles of parsimony and triage, I would focus on the Chinook recovery as something that is of the highest priority. And this morning's session was an excellent background in that, and I believe our governor of the state and his task force have that on the table. But in my mind, something has to be done before we run out of reproductive lifetime on these whales, which is only about a 25-year period, and we've been talking for 20 years, so we better get going.
Can I? Oh.
And just to follow on both of Ken and Mike's comments, it was interesting this morning to hear about the decreased marine survival of Chinook in the Salish Sea, because we are seeing a reduced habitat use in the Salish Sea by southern residents. Last year was the first time for the entire month of, so it was August 4th to September 4th, the animals were not seen in the Salish Sea. They haven't arrived yet in the Salish Sea. So if we're seeing that change in marine survival of Chinook for whatever reason, and I'll leave that to the salmon folk to sort out, that change in attendance patterns is significant for these animals because they are following the Chinook. They are foraging on the Chinook. And the second point that I want to make is, as a metabolic physiologist, we don't know for sure if killer whales are, and I would doubt they are, fasting adapted. So these gaps that are appearing in the ability and presence of Chinook for these animals might have significant metabolic repercussions that we're not necessarily seeing, but decreasing the fitness of these animals. So while we might have abundance in sort of stochastic fashion, those gaps between what they used to have as continuous Chinook availability might well be driving their fitness down as well.
Mike, did you have something?
Yeah, well, Ken started answering the other question I had in mind. But it is. Okay, I got 9 experts here. I'm going to ask a question that I probably know the answer to already, but is there a consensus amongst you on what threats need to be addressed most urgently? Ken gave his answer. Does anyone else want to say something else about it?
Mike, I think I'll chime in on both your first and your second question. I think as scientists, a lot of times we think about forming hypotheses and testing those hypotheses. One of the things we don't think a lot about is looking at efficacy of management measures that we try. And so just Jason's example of the slowdown, we were very fortunate. There was a small number of very talented people that took it upon themselves to actually study that and to see what the benefits were. And I think we're in a position now where we need to be taking actions and we need to be studying the efficacy of those actions that we're taking as we're moving forward and going along. And I would say, based on Rob's presentation and other work going on, I think we need to look at salmon and we need to look at noise combined. So that's my 2 cents.
Okay, well, I didn't ask about efficacy. That's what I was going to ask the next panel. You're the experts. You're studying these animals. You are seeing, you believe there are threats to their existence. You have a Washington State governor's panel or governor's executive order saying, sort this out, give us some answers so we can take some executive actions here. Someone has to advise them. If you need more research, then you damn well better tell them we need more, we need no more. And oh, by the way, it costs money. So you got to sort that out amongst yourselves. And if they're asking you, okay, that's all well and good, but when do these new results come online? And you tell them, oh, well, it's going to take a couple of years. You're going to have to say, these people who are trying to make these decisions have to say to you, well, we can't wait that long. The governor's pounding the table, wants an answer right now. What are you going to tell those people? This is gut reaction time on most people. You can't be strictly a scientist. And if it's, and they're not asking you to judge about the efficacy of it. That's this other group of people. What needs to be done to help recover this population? Ken's given an answer, unequivocal. There's another answer on the left as well.
Well, could I just mention, so with regard to Ken's answer, I mean, yeah, there are the policymakers both from Washington, but also at NOAA are asking a lot of questions about salmon, which salmon stocks. We're already spending, as you heard earlier, over $500 million annually on salmon recovery as it stands, compared to less than $1 million on southern resident killer whale recovery. So there's already a lot of investment in salmon. What more do we need to do incrementally? One of the scientific answers that we're trying to give as rapidly as possible to that is basically, if you're going to target increases in salmon, maybe through hatchery production in the short term, through targeted habitat actions in the longer term, what are the stocks that you want to focus on first? And we are providing answers to that question to the people who are asking that kind of right now, based on where are the whales, what salmon stocks overlap in space and time with where we think the whales are most nutritionally deficient. So that's sort of the immediate answer that we're providing to that question.
Hey, maybe one more question. Or I was going to say we probably should have a little bit of time for folks in the audience to ask questions. There's a lot of expertise there as well. But so one more comment over here. Yeah.
So my personal opinion, I mean, in the meantime, if salmon are needing time to expand and increase, as they will. I agree with the two-pronged approach about reducing noise. And I should point out that Rob and Aaron also published a paper that showed that kayaks, they respond to kayaks as well. So it's not just noise necessarily, it may just be vessels in the water. So I think just allowing them the room to be able to find the fish they need is important.
Question from Aaron, and then we'll go to see if there's any questions from the audience.
Thank you. I had a question about the impact of noise and the foraging behavior of the animals. So I reviewed the David Luceau paper as well as Marla Hood's DTAG data papers, and neither of them seemed to analyze whether boats that had echo sounders operating had a disproportionate impact on the behavior versus either kayaks or boats without. So I was wondering, could someone summarize, has that question been examined about the effect of echo sounders on or off? I think Rob Williams would be the natural person to ask this to.
I'll take a crack at that, Aaron. We haven't specifically looked at echo sounder effects on their foraging efficiency. I can say that actually that one of those curves, that the black curve, the cumulative distribution curve on that plot right there is actually 50 kilohertz echo sounders at lime kiln. So they're prevalent, and if you sample higher, you'll also get the 200 kilohertz depth sounders as well. So it's prevalent in these small boats. Our behavioral response and echolocation click masking models to date. Actually, if you flip to the next slide, Robin. It's a convoluted model that I can't get into right now. I can explain it later if you want. We can have a discussion. But based on summertime with whale presence in the Salish Sea, the central Salish Sea area, we're predicting that there's about a 20% loss of foraging time between a combination of behavioral response from acoustics from vessels and echolocation click masking, if you combine them into a simple noise metric. And roughly about 2/3 of that lost foraging time is due to ship noise, and 1/3 is due to small boat noise, so your small private boats and whale watch vessels.
Thanks. I think we have time for one or two questions from the audience, if anyone has questions. And if you do, please use a microphone and state your name and affiliation. I will also point out that in the next session, we'll have another panel discussion, which will share a couple of the panelists and some opportunity there as well. So, Sam.
Obviously, acquisition of resources is important, but transfer of the resources to the calf is also important. And I didn't notice in some of the activity budgets, what do we know about nursing, periods of nursing, when that's important, how that might be affected by any of the vessel or other disturbance activities?
That's a really great question. It's very difficult to observe that because the water is not that clear. I don't know if you can see better from the theodolite, but it's hard. Even if you had a theodolite study, it's a very static place. They have to swim by and then they're gone. And so unfortunately I don't really know how to get a handle on that. And there's been relatively few calves spread across the year, so sample size would be small.
Yeah, it is a great question. Dawn and I have published on the energetic cost of lactation with respect to mom's nutritional demands. What we could do is go back through some of our older data and look at inter-individual spacing. And I have not seen anyone look at that to see whether, as the number of boats increase, the spacing among individuals increases. But that we could probably do that as proxy for nursing. I'm aware, though, of Michael's question, which was a really important one. Are we doing all the science that we're doing? And I think one of the things that I'm hearing is that there's a lot more going on than I knew about. And it's great that I'm hearing about work that Sheila and DFO are doing. It's great to hear about all the work that the aquarium is doing. I think I'm old enough to remember when the Americans had all the money, you know, and it is wonderful to see how much money Canada is spending on science. I think to answer your question, it may well be that all the main threats are being addressed, all the main demographic data gaps are being addressed. But there is certainly a need for a coordinating role, and there certainly is a need for better communication among government, academic nonprofit independent scientists all working on these issues together. I think working in a transboundary space, our nonprofit is registered in both Canada and the US, foreign vessel clearance, trying to have permits in both countries. I think we could do a lot better. We could do our science more efficiently, and we could share information, and we could take advantage of a whole lot of unpublished data sets or repurpose old data sets that don't currently exist in one place. And I think we're losing institutional knowledge, and we run the risk then of duplicating studies that have been done before. Thank you for reminding me of that one that we did with David Lusso that never did get published. I'm sure we each have a handful of those that are worth kicking out the door.
Well, I also think there was some northern resident DTAG work done a number of years ago, and you wonder about bringing those older data sets into the current questions. Be really valuable. I think we're running up just over when our break is supposed to start. I don't know if there's any last questions or comments from the Commission. No? Well, everyone, if you could join me in thanking the panelists and the speakers. It was a really useful discussion. So.